HIV and monoclonal antibodies, AQA biology. Key terms: polyclonal antibodies, which are antibodies that can bind to many antigens, and then monoclonal antibodies is when antibodies can only bind to one antigen. HIV. AIDS comes from HIV. And the origin of HIV is basically an infection occurred in Central Africa around about the 1930s. And it jumped the species barrier due to eating or slaughtering chimpanzees or monkeys. How does the virus infect? Well, it's infected by sexual intercourse, drug use, blood infections or wounds, blood transfusion/products, breast milk, but not direct contact, honestly. Like, you could shake hands with someone that had AIDS, it's fine. Or HIV. HIV prevention: safe sex, avoid promiscuity, don't share needles, screen all donors and products, extreme care when dealing with known AIDS sufferers, and HIV testing of all partners. So, how is HIV diagnosed? Well, you use an ELISA test. And what are the different causes of HIV? Well, you have the acute phase, in which you get fever, sweats, and headaches, and this lasts for about 3 to 12 weeks. And it's rapid viral multiplication and loss of helper T cells, and you start to feel better. Then the chronic phase can last up to 20 years. The virus produces infected T cells and are kept in check by, yeah, so it's kept in check, sorry. Active virus presents, so HIV is infectious, and there's 10 to the power of 9 viruses produced daily. And you have a reduced immune system, and TB or shingles may be active. Now, during the disease phase, this is basically when people get AIDS, and they get AIDS when there are less than 200 helper T cells, and the occurrence of any more than 20 infections. This is the structure of HIV. So you have attachment proteins, which are here. Then you have your genetic material, which are here. Then you have a capsid, which is this. Then you have your reverse transcriptase, which is these little circles. Your lipid envelope, which is here. And then you have your matrix, which is this area. So why are there no vaccines? Well, this is because vaccines mimic real-life infections, and the body makes antibodies so the pathogen in the future is destroyed. But HIV hides in helper T cells, so it can't be destroyed. And there are different strands of HIV, so you're going to need different antibodies to fight the different strands. So why no antibodies? Well, antibodies can only fight bacterial enzymes, and viruses use host cells. So if you want to get to the virus, you'd have to kill your body cells. So how is it treated? Well, you use AVRs, in which insert yourself into the DNA of the host cell, and then it switches thymine with AZT, and this causes the slow replication of the virus therefore the destruction of the helper T cells due to reverse transcription. And reverse transcription catalyzes the production of DNA from RNA. Monoclonal antibodies. How monoclonal antibodies are made? Well, you mix a cancer cell with the B cell, and they mix to form a hybridoma cell. These divide, and these produce monoclonal antibodies. Detergent plus B cells plus tumor cells. Well, you use detergent because it breaks down the cell membrane, allowing cancer cells and B cells to fuse. Monoclonal antibodies have been made into living cells because otherwise the body will reject it. It will identify it as foreign. What are the 4 ways that monoclonal antibodies can be used in science and medicine? Well, you have pregnancies, ELISA test, cancer treatment, and haemophilia. So how does the ELISA test work? Well, you have the monoclonal antibody that's coated well, and then the antigen binds to the antibodies. A second monoclonal antibody linked to an enzyme binds to the antigen, and the substrate is added, and this substrate added causes the enzyme to change color. So the more color formation is the more of the antigen that is there. So cells from cancer cells used to form with B cells. Well, they're used because cancer cells are rapidly dividing cells, so you can get a lot of antibodies formed in a short period of time. And monoclonal antibodies need to be humanized so that the body doesn't reject it and it can create an immune response. So hopefully you liked this video, and that's all there is about this topic. The next video will be about exchange. So if you like the video, please like and subscribe. And thank you for listening.